Hello, everyone, and welcome to today's webinar. This is sponsored by Chantest, but we have three great speakers who are going to share with you lots of information today. The title of today's webinar is Dialing for Targets, Procuring Cells, Expressing Channels, and Receptors for Biotech Research. My name is Dawn Van Dam, and I'm the General Manager of Cambridge Tech Associates, and also the Manager of the Drug Safety Executive Council. I will give you just a quick overview of today's presentation and then introduce our three great speakers for today. We have three different segments where we're going to be discussing the use of cloned cell research. We're going to be looking at the evaluation of the mechanism of action of allosteric modulators and its application to drug discovery. We will be looking at label-free solution assays in the study of orthosteric and allosteric GPCR ligand binding using backscattering interferometry. And the third presentation will be dialing for targets, procuring cells, expressing channels, and receptors for biotech research. So to introduce our speakers for today's presentation, Luke Armstrong is the Director of Cell and Molecular Biology at Chantest. He earned his PhD in biochemistry and molecular biology at the University of California, Davis in 1994. He then did his postdoctoral studies at the University of Washington. Before joining Chantest, Luke worked as the Manager of Research and Development at EMD Millipore. Our second speaker is Richard Jake Isaacs. He received a PhD in biochemistry studying the structural biology of carcinogen damage and base mismatches in DNA. He did postdoctoral work at Vanderbilt University studying lipid antigen-presenting membrane proteins and development of a qPCR assay for identifying and quantifying pathogens in the lungs of critically ill patients. He is a former scientist or senior scientist at Microbial Novatech, developing and deploying a high-throughput cell-based assay to identify viral protease inhibitors. Currently, he is the Applied Research Laboratory Supervisor for Molecular Sensing, overseeing drug discovery services projects for major pharmaceutical clients, evaluating the backscattering interferometry molecular interaction platform. And our first speaker today, I believe he will be going first, is Jeff Reagan. He received his PhD in chemistry from Yale University in 1986. He pursued postdoctoral studies at the Yale University School of Medicine in the Department of Neurobiology, studying the molecular pharmacology of olfaction. He joined Sandoz Novartis in 1989, working in the area of G-protein coupled receptors. In 1998, he joined AFMAC, where he extended his work on the molecular pharmacology of GPCRs. In 2001, he joined Turlic, acquired by Amgen in 2004, and has worked in the area of diabetes, nephrology, and cardiovascular disease, with a specific emphasis in systolic heart failure. His key area of interest is the molecular pharmacology and mathematical modeling of allosteric modulators. So, with no further ado, I will turn it over to our first speaker, who looks like actually it's Luke who's going to go first. So we'll pass the ball to you, Luke, and give you an opportunity to share with everyone what your information is today and provide your presentation. But before I officially turn it over to Luke, I just want to remind all of our participants today of two things that we always do. One is that we ask you to submit your questions in the chat box, and then I will ask the questions of the panel. And then the other thing that we always do is we post a survey at the end to get your feedback on future topics and other things that you'd like us to consider for future webinars. So please be sure to open up that survey before the end of the webinar and fill it out for us. So with no further ado, I'll turn it over to Luke, who is our first speaker for today.
Thank you very much, Dawn, and thanks to the listeners for attending. I'm going to start the session by telling you about processes that we use at Chantest to develop and provision cells expressing recombinant receptors and channels for biotech research, and this is both for internal use and as a service to our customers. And I'll pay particular attention to preparation of cellular membranes that can be used in binding assays that Jake and Jeff are going to tell you about. So first, I'd like to give you some background about Chantest. Chantest was founded in 1998 by Buzz Brown, who's pictured here, and to provide a service to predict drug-induced cardiotoxicity that's caused by inhibition of the hERG potassium channel. And the company has grown to over 50 employees and has expanded its scope quite a bit. So we now have several service units. So the safety testing unit is the first one that was founded, and still focuses primarily on hERG and other cardiac channels, and it's still the backbone of the company. But we also offer discovery services for projects requiring high throughput screening of ion channels and GPCR targets. And finally, we offer transporter services, primarily for CFTR, for cystic fibrosis research. And we do offer reagents, and for most of the services that we run with the ion channels and GPCRs, we offer the corresponding products. And finally, we offer cell culture services that are tailored to the needs of the individual customers. And we're proud that we've been named the most trusted fee-for-service provider since 2006 by the HTS Tech Ion Channel Survey. So this slide just shows how the Cell Culture Services Group at Chantest serves both internal and external customers. Starting at the top left, one of our key functions is to provide ion channel and GPCR cells to our safety and discovery services units, and we do this on a daily basis for those units to run their studies with customer compounds. And another function that we perform is to develop new cell lines for the safety and discovery units at Chantest. And we are happy to develop new cell lines on a custom basis for external customers. So in addition, we maintain our reagents portfolio, which contains most of the cell lines that are used in our studies, as well as some single-use versions of the cells and membrane preparations from those cell lines. And finally, we offer cell culture services. This includes custom scale-ups of cells and membrane preparations. And we can do this both for our own cell lines and cell lines that customers provide. And we do use the same methods that we use for banking cells for our own internal use for the services. So going back full circle here. So I will cover the cell culture services that we offer and start off with the development of stable and transient cells expressing ion channels. So our stable cell line development service encompasses molecular cloning, transfection of the plasmid, selection of stable integrants, and cloning those out into single cells, and then selecting the clones by functional assay, either on automated patch clamp or by fluorescence assay. And the clones are then validated functionally with full pharmacology done, and also at the molecular level for expression. And our team does have extensive experience in cell line development with over 80 ion channels and over 70 GPCR cell lines under our belts. So the platforms, this slide just shows the platforms that we use for analysis of our ion channel cell lines and GPCRs. At the top is the Q-patch from Sophion, which performs automated whole cell patch clamping. And at the left is the IronWorks Barracuda from Molecular Devices, and this performs perforated patch clamp, and it's preferred for higher throughput applications. And on the bottom is the classic manual patch clamp method. This is still the favored method for safety testing, although it's the lowest throughput of our patch clamp techniques. And finally, for discovery purposes, we use the flipper from Molecular Devices to perform calcium and membrane potential assays. So development of cell lines, especially for ion channels, can be tricky, and there are a number of hurdles that we are equipped to surmount here. And so this slide just illustrates one common problem with development of any cell line, and that can be loss of expression. This cartoon just shows one mechanism for this, where the DNA around the integration site becomes methylated, chromatin becomes compacted, and the transcription is silenced. Another issue to contend with in ion channels and GPCRs is that of trafficking to the cell surface. Ion channels and other membrane proteins transit through the secretory pathway, and once upon the cell surface, also undergo internalization. And if the channel becomes sequestered in any of the various organelles in the interior of the cell, this can lead to reduced cell surface expression and impaired signals. So ion channel trafficking, interestingly enough, has practical importance because some compounds can interfere with hERG trafficking and can behave like direct hERG inhibitors and can contribute to cardiac arrhythmias. And we have an assay called the hERG light assay that tests for this specifically. So another big factor to consider in ion channel functional assays is what we call patchability, and that's the ability to configure the cell properly to obtain and then maintain seals in the patch clamp application. And this is a cartoon that just illustrates the inner workings of a generic automated patch clamp instrument. There is a chip that contains micron-sized pores that's positioned in the instrument, and then the cell is suctioned onto that pore, and the membrane is broken and at the site of the pore, and then electrodes that are positioned in the intracellular and extracellular solutions measure the current that's generated by the channel. So you can imagine that any abnormalities in the shape or in surface contours of the cell can affect the positioning of the cell at the pore and the efficiency with which it seals onto the pore. And then also, if cells are aggregated, that can impair detection of the current as well. And then finally, the cell needs to remain healthy even though it's got a hole poked in it for several minutes while the reading is occurring. And so to optimize the performance, we deal with expression issues by subcloning or cell sorting. We manage issues of poor trafficking by temperature. We can incubate cells at a reduced temperature prior to the assay to improve trafficking to the cell surface and improve signals. And patchability is usually dealt with by modifying cell detachment processes and culture conditions. And we regard these parameters as being essential components to our cell line development service. And optimizing this is not a trivial matter, and so we've got a lot of expertise in that. So an alternative to the development of stably expressing cell lines is performing large-scale transient transfections. And we do that internally and as a service as well. And so our transiently transfected cells, we call EZ cells TT. They're transfected with an ion channel cDNA and are designed for single use and by the customer and can't be propagated. In some cases, the method does have advantages over replicating cell lines. That includes faster turnaround time from the gene to the assay. There's a reduced cost for smaller projects, and then the ability to express multiple subunits without having to culture the cells like stable cells in a cocktail of toxic selection agents. And so the figure here just shows the MaxCyte large-scale electroporator that we utilize, and we can make up to 300 million cells in a run. So once you have a whole bunch of cells, either stable or transiently transfected, it's most convenient to have cryopreserved large banks that you can use at your own desire. And so we offer a service to prepare large-scale cryopreserved cells. So we can prepare the cryopreserved cells at up to 10 billion cells per batch, and to grow this many adherent cells, and we almost deal exclusively in adherent cells, we rely on several types of multi-layered culture vessels. And we utilize a step-down freezer, the cryomed, down below on the bottom right, to accommodate the large numbers of vials and also to give reproducible freezing conditions. And finally, shipping is not a trivial matter when you have a huge bank that you want to keep happy. So we ship our larger batches of cells at liquid nitrogen temperature with the cryoport shipper. So we can do various kinds of projects, including large quantities of one cell line over smaller batches of panels of cell lines. And finally, we can scale up cells for membrane preparations, and that is the final topic of my talk here. So we can make batches of up to 2 grams of membrane protein, and we do have flexibility in the protocol that we use depending on the needs of the customer. And there are a couple methods for disrupting the cells, nitrogen cavitation and homogenization that we use. And also we can formulate the cells however best works for the customer. And for example, the backscattering interferometry technique that Jake will tell you about next works better without some of the carriers that we usually put in there. So we perform QC on our membrane preparations by radioligand binding assay, and this figure shows analysis of membrane preps from cells that are expressing the M4 muscarinic acetylcholine receptor, a GPCR. And on the left is binding of the radioligand N-methylscopolamine, and on the right is a GTP gamma S binding assay, which assesses activation of the receptor. One limitation to such radioligand assays is that the availability of radioligands can be kind of limited, and that's one of the reasons why molecular sensing's technology that Jake will tell you about next is so exciting, because it frees you from the need to use radioligands. So to conclude my part of the presentation, I'd just like to offer Chantest services to customers who are needing cells that are expressing channels and receptors. We have a very experienced team with modern instrumentation, and we're accustomed to delivering our services on a very tight timeline. So with all of our experience in ion channels, GPCRs, and transporters, we have become a trusted provider in the field. And so we at Chantest are ready to meet your needs for cells expressing ion channels, GPCRs, and transporters. And thank you very much, and I will hand the floor over back to Dawn.
Great, thanks so much, Luke. And again, Luke is the Director of Cell and Molecular Biology at Chantest, and that was Luke Armstrong. And our next speaker will be Jake Isaac, and he is with Molecular Sensing, and he's the Applied Research Laboratory Supervisor. So Jake, if you just move the little, yep, perfect, great, thank you.
Thank you, Luke, sort of priming the audience for what I'll be speaking about today. And of course, thanks to all the attendees for your time today. So I'll be providing some background on what's probably an unfamiliar technology to most of you, and that's our proprietary backscattering interferometry, or BSI platform, and specifically how it's been applied to what's a particularly challenging but very high value class of drug targets, the integral membrane proteins. Okay. So this is just a very basic schematic of how backscattering interferometry works. The core of the system is this microfluidic chip, and samples are passed into this chip and flow is stopped, and the helium neon laser is directed onto the channel. Within this channel, there are a series of internal reflections and refractions that result in the rays being reflected in the opposite or back scattering direction after interacting with the sample. And these rays, or interference pattern, is captured by a CCD array or CMOS camera. And what that pattern looks like is sort of depicted in the upper left here. And the reduction into a more one-dimensional representation is depicted here. So we capture these images, and essentially any movement of that pattern that we can interrogate in various ways with some proprietary signal processing algorithms would indicate a change in the sample. That could be a refractive index change, or a change in the sample that's indicative of conformational change in the target induced by ligand binding. So the way we exploit this is typically we set these up very similar in protocol to the way you would do a radioligand binding assay. So we have a discrete set of samples mixing drug target with your drug of interest, so that when the drug interacts with the target, there's a conformational change that occurs. So there's a fundamental difference in the BSI signature of a unbound versus a bound protein. So we set these up such that the drug target is held constant and a discrete set of samples, typically a 10 to 12 point titration curve is constructed with the drug. The drug or its buffer tends to have some refractive index of its own. So we subtract that out of the overall signal of AB, we refer to as our assay channel. And the difference curve is what we use to compute the dissociation constant, or KD, for this system. So we've probed how BSI signals are correlated with conformational change. And this is a sort of control experiment we've done a number of times, where we look at the BSI signature of bovine serum albumin at various pHs. Now, it's known from various orthogonal techniques that BSA undergoes a series of conformational changes as you change pH, going from open to closed conformations. And comparing this with a densitometry study that was published in the Biophysical Journal in 2006 by Wax et al., this measures the overall volume of the BSA at these various pHs. And you can see there's a great degree of concordance between the BSI and the densitometry. And we've done a number of other experiments as well to validate this mechanism of detection. So currently, we perform our drug discovery services with a pre-commercial prototype. Our current generation is a dual-channel assay instrument, so that we have our assay and reference samples drawn in by an autosampler simultaneously to control for very small fluctuations in temperature and other conditions. The autoinjector is fully robotic and compatible with 96 wells and in the future 3 to 4 wells. A commercial version of this system will be available this fall. So one of the major strengths of the BSI platform is its ability to probe interactions between very complex targets and small molecule drugs. And we're able to look at integral membranes in a variety of different types of preparations, ranging from physiological to more artificial. We can look at small ulolaminar vesicles that are produced by ultrasonication of cell pellets. We can look at one extra step of purification, the crude membrane preparations. This is overwhelmingly the form that we tend to look at integral membranes in for our clients as well as third parties. We've also looked at lipoparticles. These are produced by co-transfection of your target of interest with a viral construct, such that you get these virus-like particles that bud off of your cells. And we've also looked at nanodiscs, which are slightly more artificial but very stable way of expressing membrane proteins. Typically, your protein is purified from a detergent solubilized cells. These are mixed with lipid and a amphipathic structural protein. The detergent is dialyzed away, and these self-assemble into a bilayer with your target of interest contained within it. So the remainder of the sort of data slides will be concerned with the M4 muscarinic acetylcholine receptor, and these we studied. The material was acquired from Chantest, and as Luke said, there can be definitely an effect in our assays on the quality of the membrane preparations. We found that the nitrogen bond technique produces a very nice, clean membrane fraction that's very amenable to BSI analysis. So just a little background on the M4. It's a very important drug target in the central nervous system for a variety of conditions, and is it's a prime target, especially for Parkinson's. So we looked at the binding of the endogenous agonist acetylcholine, as well as a positive allosteric modulator that's commercially available, this VU compound. So this just shows a direct binding assay using BSI for acetylcholine against the M4. Now, one of the major strengths of BSI is immediately obvious here. We can look at very small molecules binding to very large drug targets, such as M4. This can be a challenge for other technologies, such as surface plasmon resonance, where there's a mass dependence, and it's quite difficult to get accurate binding constants for small molecules binding to large targets, such as integral membrane proteins. Now, a very interesting type of experiment we can do is looking for cooperativity between molecules binding at different sites. In this case, we're looking at binding of the acetylcholine with and without the positive allosteric modulator. You can see that when the allosteric modulator is added into the samples, you get a dramatic shift in the KD of the acetylcholine. It's about a 20-fold shift in the KD. Now, you can also do sort of the reverse experiment. This is just showing the binding of the positive allosteric modulator itself. And again, this demonstrates a core strength of BSI. You can look at the binding of various molecules in a binding site agnostic sort of way. You're not limited by having a radio-labeled compound that you're looking for direct binding of or competition for your test molecules. You can look at direct binding of allosteric modulators in without any labels whatsoever. So we can see when we do the similar experiment, looking at binding of this positive allosteric modulator with and without saturating amounts of the acetylcholine, we can again see a dramatic shift in the KD, and again, the shift is about 20-fold tighter. So when you compare these two results, what you want to see is an alpha or cooperativity factor that's fairly similar when you sort of flip the order in which the mode in which you analyze the binding of these molecules. So that's really all I wanted to present today. Again, thanks to all the speakers and attendees today, and I'll be happy to take any questions. Or are we taking those after the end of the talks?
Yes, we will, Jake, at the end of the talks do questions. So again, I will encourage everyone to submit their questions. And we'll move the slide along to our next and final speaker for today. We have Jeff Reagan, and he is Scientific Director at Amgen. So over to you, Jeff.
Oh, thank you very much. So my talk will really complement the discussion that Jake just made concerning allosteric modulators. And I'd like to go a little bit into some of the concepts related to allosteric modulation and then to get to some of the data. So the data I'll be showing you today is actually generated by molecular sensing. We at Amgen have a strong relationship with molecular sensing. We have a contract with them to do some binding studies. And we're going to be the first company to receive one of their interferometers later this year, and I'm very excited about that. I've already got a number of groups at Amgen that are interested in exploring this technology. So I'm a big fan of the technology, and so let's move ahead. Next slide, please. So as Jake alluded to, allosteric modulation is an important aspect of drug discovery, and we at Amgen are very excited about this area. We have a drug called Sensipar in the market that is an allosteric modulator to the calcium sensing receptor, and I just wanted to give you a little bit of the jargon that's used. You'll hear two terms used, orthosteric and allosteric. And all this means is that the orthosteric site is the site to which the natural ligand binds, for in this case that Jake discussed with acetylcholine, which I've depicted here in red. The allosteric site simply means different. It binds to a site that is distinct from the orthosteric site. And what's important to understand is that when you have an allosteric ligand and an orthosteric ligand bound simultaneously, they can each influence their binding properties. Next slide, please. So the way we set up our experiments that I'll be showing you with molecular sensing is we first look at a receptor, which I've shown here in blue. with the orthosteric ligand shown in red, and we generate a binding curve and a dissociation constant, which we refer to as Ka. That is the dissociation constant of ligand A. Next slide, please. We then add to that, after we have saturated the receptor with ligand A, our allosteric ligand B can further achieve equilibrium to the receptor ligand A, ligand B complex, which we refer to as RAB. Now this affinity we refer to as Kb over alpha, the dissociation constant of ligand B, divided by a cooperativity factor alpha. So if alpha is larger than 1, that means that B decreases the affinity of ligand A to receptor. If alpha is between 0 but less than 1, it increases the affinity of ligand A to the receptor. Next slide, please. But you could also envision doing this experiment in the opposite fashion, in which ligand B is added to the receptor to establish the affinity, Kb. Next slide, please. And then add ligand A, and again establish equilibrium to be the RAB complex. And here we've shown that as Ka over beta. So the question one might ask is, what is the relationship between the cooperativity factors alpha and beta? Next slide. And it turns out, as Jake alluded to, they should be equal. And the reason for this is because this is a closed system, and under equilibrium conditions, the Gibbs-free energy change from going to R to RAB is independent of path. So it doesn't matter if you add ligand A first or ligand B first, you'll get the same cooperativity values of alpha equal to beta. Next slide, please. So as Jake just alluded to, we're using a backscattering interferometry, which I won't go into because Jake just discussed that. Next slide. So the first experiment we did was with a receptor, which is an intracellular soluble enzyme complex. In this particular case, we're able to do the backscattering interferometry with soluble proteins as well as membrane-bound proteins. This particular receptor is as I said, is a soluble ATPase. And what we're doing first is we're adding our drug. So we refer to our drug as B, and the natural ligand to this target is A. So when we establish an equilibrium between this intracellular receptor and our allosteric ligand B, we achieve a nice binding curve here with an apparent affinity of around 300 nanomolar. Next slide, please. The next experiment we do is we fully saturate the receptor with ligand A, with the receptor, the concentration at A being about 50 times higher than the dissociation constant, so it's pretty much fully bound. Then add ligand B, and again establish an equilibrium where we can then query the cooperativity value alpha, shown here 30 nanomolar. Next slide. Oh, that slide, correct. And what you can see is we see a nice left shifting of the dose response curve. That is to say, in the absence of the natural ligand, we get an affinity of around 280 nanomolar. In the presence of the natural ligand, we get an affinity of 30 nanomolar, giving a cooperativity value of approximately 9. Now this is very important for us because we have some emergent hypotheses concerning the importance of alpha on the pharmacodynamic effects. Next slide, please. This is the second molecule that we've tested that we call compound D2. And again, what you can see here is it has a similar cooperativity value of about 4. So in the presence of ligand, which is shown in blue, of natural ligand, we get an affinity of about 40 nanomolar. In the absence of natural ligand, shown in red, we get an affinity of around 150 nanomolar. Next slide, please. And this is the third ligand I'd like to show you. This is compound B3. And again, we're seeing this left shifting of the curve. So basically, what we're seeing here, next slide, please. When we examine these three compounds, notice that the cooperativity values we're seeing are not that far off. They're both less than, all three of them are less than 10. And what we are interested in doing is combining this information with functional data that will provide us important clues as to how our medicinal chemistry efforts will go forward. With this particular target, we are seeing some interesting additional effects that we can't fully explain, and we're querying whether or not this value of alpha has anything to do with these additional effects we're seeing. I might also add that we do not have the ability to radio-label either ligand A or B, so we really can't do any kind of binding assay. So this methodology is, I think, really spectacular for being able to query this particular protein, which we don't have any other methods to do. We've also investigated BioCore technology, but we have not been able to take our target and attach it to a chip such that it doesn't destroy enzymatic activity. That's one of the big problems with BioCore that we have found, is that oftentimes when you try to attach your target to a chip, you lose activity. With this particular technology, it's label-free. You just mix everything in the cell and you go. So we're very, very impressed with it. Next slide, please. Now, the one question you might ask if you have done some experiments with radioreceptor binding is, what about nonspecific binding? How does that play into the equation? And what I've shown here is a binding assay with some Chantest membranes we actually purchased using N-methylscopolamine. And what you can show here is the total binding shown in black and the nonspecific binding. And the way nonspecific binding is done in a traditional binding radio-labeled assay is to include a 1,000-fold excess of cold ligand, so you can get just the nonspecific sites. And then you subtract total from nonspecific to obtain your specific binding. Next slide, please. In the case of backscattering interferometry, you don't have a nonspecific binding curve because there is no hot ligand to add. Basically, you just simply are, as Jake alluded to, you are subtracting out the background from A without receptor. So the question is, why don't we have a nonspecific binding signal? Because you'll notice on the right-hand panel, the drug-receptor interaction is saturable, indicating you're binding all the sites. Next slide, please. So what we think is going on here is that backscattering interferometry is really querying big conformational changes in the protein. Next slide, please. Next slide. Yes. And nonspecific, previous slide. And the nonspecific binding, we don't expect to be a very big conformational change. It's typically very low affinity, typically 1 micromolar or greater. And we don't expect the binding to a nonspecific site to impart a big conformational change. So we don't expect to see that, and that's pretty clear from the right here that we don't have the issue of nonspecific binding. And I cannot overemphasize this point, in that I've been doing binding assays using radioligands for upwards of 20 years now. And the biggest problem I've faced with doing radioreceptor binding assays is that organic molecules are typically very hydrophobic, leading to very large nonspecific binding curves. And that's not an issue with backscattering interferometry. So I see this technology as being very useful for looking at, in particular, hydrophobic ligands binding to both membrane-bound proteins as well as intracellular proteins, as I've shown you here. And my 10 minutes is almost up, so I think I'll stop there and turn it back over to Don.
Great, thanks so much, Jeff, for the great presentation. Okay, so we're open for questions, and I have a couple of questions that have come through already. So maybe I will turn first to Luke. I'm just going to go back to there we go, so everyone's picture is right there. So Luke, a question that's come in for you is, how do transiently transfected cells compare to stable cell lines in terms of signal window and variability?
Well, the transiently transfected cells tend to be a little bit lower in the signal window and in the variability as we assess by Z prime, but we do have standards for the transiently transfected cells that each batch has to pass, and these are screening quality as well. And I should emphasize that we use transiently transfected cells in favor over stable cell lines for some targets. For some targets, it just seems to work better, and it seems to be ones that are where there's some toxicity associated with the channel. So.
Okay, great. Well, thank you very much for that answer. That was really helpful. So I'm going to ask the next question of Jake. And again, I'll remind everyone to please put their questions into the chat box, and also to download the survey that's in the chat box link and just start to answer those questions too. So question for you, Jake, is how is BSI different from other label-free biosensor technologies, such as SPR and grading coupled approaches?
I would say probably the main advantage is the removing the requirement for tethering your target to the surface, and also removing any mass sensitivity you might have. As I pointed out, you can look at very small molecules binding to very large targets. Also, without having to tether to a chip, you can dramatically simplify the assay setup and development process. Typically we can have a functioning assay within a week or two. So I'd say that is probably the primary advantages.
Okay, great. Well, thank you very much. The next question I'm going to ask of Jeff. So Jeff, how does the cooperativity values you obtain from the binding data compare with functional data?
That's an excellent question, and we're actually seeing a discordance currently with this drug series, in that where our functional data indicates we're left shifting the curve by about 1.5 to 2 orders of magnitude, whereas in a binding assay we see less than 1 order of magnitude shifting. So what that's telling us is that the binding properties of the drug are mainly influencing some downstream effect other than the binding affinity of the natural ligand. So if the cooperativity for a binding assay and the cooperativity for the functional assay were approximately the same, then one could explain the mode of action of the drug as being primarily affinity modulation. If there's a discordance, we have this concept of what's known as efficacy modulation, in which there's some other factor involved besides the interaction between ligand and A and B going on. And that's what we're seeing in our particular series, is that there's this discordance. So the affinity modulation only partly explains the mode of action of the drug and not fully explains what we're seeing in terms of the full functional effect.
Okay, well, thank you very much, Jeff. That was very helpful. Another follow-on question for you, Jeff, is what is the throughput of this assay?
Currently, we're able to do several molecules a week. My plan is that when we receive this instrument in September or October, I plan to be able to run probably about 6 compounds a day would be our hope. That would be sufficient to support our SAR efforts. I don't envision this machine being a high throughput instrument. I see it as an instrument to support SAR efforts where we're typically making anywhere from between 15 to 30 to 40 molecules per week in which we want to obtain binding data.
Okay, great. Thank you very much. I'm going to turn it back to Jake for the next question. Jake, how difficult is it to work up assay conditions for BSI analysis?
Yeah, I believe I partially answered that in the last question. It's actually quite simple, mainly due to the free solution aspect of the technology. We describe it as a mix and read sort of technology. You can mix samples together, target and ligand, allow them to equilibrate for, say, a couple of hours, and then start injecting them into the instrument. The BSI instrument is tolerant to a wide range of buffer conditions, DMSO concentrations, and other additions to the buffer system. So yeah, it's actually quite simple to work up an assay in the majority of cases.
Okay, great, thank you. So perhaps a follow-on question for you, Jake, is why can BSI succeed in the analysis of difficult to analyze targets, such as authentic membrane fractions containing targets of interest?
Yeah, I think yet again that's kind of an aspect of the free solution technology. You can have proteoliposomes such as you get from crude membrane fractions, as long as these are stable and in suspension or solution. It makes it fairly amenable to look at these complex targets. We've looked at a range of different types of complex targets, including multi-subunit complexes, integral membrane proteins, and a variety of different conditions. In almost every case, we're able to really produce a nice, robust assay. So yeah, this all makes BSI really amenable to looking at these very, very high value but typically technically challenging targets.
Okay, well, thank you very much. I really appreciate that. And again, if anyone has more questions, we would certainly love you to submit them in the chat box. So a question for you, Luke, is someone is saying here, we have used Chantest assay services in the past and are interested in doing some similar assays at our own company. Can we obtain the same cells that were used for the services?
Yeah, actually, yes, and we do provide the same cells on a regular basis, and this is, you know, typically for the cases where we use translinearly transfected cells. We also provide the translinearly transfected cells to our customers as well, so that they can perform experiments in parallel to the ones that we're performing.
Okay, well, that was really helpful. Thank you for that answer, Luke. Jess, another question for you here. Have you compared the binding properties of this protein using the BioCore technology?
Well, as I said, we have not been able to perform BioCore studies with this particular target. Because Amgen's a protein production, has a lot of protein drugs, we are heavily invested in BioCore technology. And we have used that for looking at antibody-antigen interactions, where it's a straightforward process to attach your receptor to the chip. However, in the case of GPCRs and intracellular targets such as I'm working on, that technology has not been amenable because attaching the chip to the receptors is problematic. It results in an impairment of activity. So we haven't been able to use that. On another point, as Jake mentioned earlier, looking at small molecule protein interactions is problematic with BioCore because BioCore looks at mass changes between the protein and the protein ligand complex. And since small molecules typically have a molecular weight of less than 500, oftentimes those mass changes are so small you wouldn't see them with BioCore. So I see these technologies actually complementary. BioCore you can obtain you know, on-off rates as opposed to just equilibrium constant. But again, the problem with the BioCore is I can't really compare it to my system because I cannot make a chip attached to my receptor in a functional form.
Okay, well, thank you very much for that. I have another question for Jake. So Jake, is the BSI technology appropriate for HTS, and any experience examples to share?
Yeah, that's a great question. We get this fairly often. As Jeff mentioned, currently the throughput is really not amenable to HTS with the current generation of the instrument and the technology. That is a direction of development. We'd like to increase the throughput of the instrument, decrease sample size, and some other directions. Currently, I would consider it more a secondary screening type instrument, and not so much HTS.
Okay, great. Well, thank you for that answer. I'm going to give another call, last call for questions. Again, if you could put them into the chat box and send them to, like you just choose my name, send them to Don Van Dam. That would be really helpful. And while we're waiting to see if there are any additional questions, I'll just go around the presenters and see if there's something else that you would like to mention as sort of concluding comments. So I'll start with you, Jake, since you were the last person to speak, and we'll go from there.
Sure. It looks like I got a question about any specific experience or examples to share. I'm not sure if that was in relation to the HTS question or in general, but I can say that we've looked at a variety of different types of integral membrane proteins, receptor tyrosine kinases, GPCRs, ion channels, and some others. In addition, we've looked at a wide range of soluble proteins, kinases, transcription factors, really any system where there is conformational change or some other change in, say, solvation around the binding site of a target should be amenable to BSI.
Okay, great. Well, thank you for that. Jeff, how about yourself? Any concluding comments from yourself?
Sure, I'd be happy to do that. One of my main functions at Amgen is to play a supporting role for chemistry, for medicinal chemistry. And if you talk to medicinal chemists and you ask them, what's the most important thing you want to know about your molecule, and they'll tell you, what's the binding affinity to the target? And as I've mentioned to earlier, sometimes that's not a straightforward thing to obtain with other technologies. And as I said, this molecular sensing technology, the backscattering interferometry, essentially allows me to answer that question for both soluble proteins and membrane-bound proteins. So our chemists in particular are very excited about this technology because it answers a fundamental question that they have about their compounds that they can often not obtain via other methods.
Okay, well, great. Thank you very much for that, Jeff. Sure. And then final comments from you, Luke?
So actually, I do see a question from an attendee here about what is the level of expression of membrane protein in membrane preps that hasn't been answered yet. And so I can answer that it really depends on the channel or the GPCR that you're looking at. And in general, ion channels tend to have less expression than GPCRs, and we measure our expression in terms of picomoles per milligram is determined by radioligand binding assay. And for ion channels that we have, usually from about 0.5 to 2 picomoles per milligram, and GPCRs can be 1 to 50 picomoles per milligram. And yeah, I think that wraps it up for me.
Great. Well, thanks for taking that last question, Luke. Certainly appreciate it. Was there anything that you wanted to say as closing remarks at all?
Just that we at Chantest have a lot of experience with functional ion channel assays, and I think that there's a lot of complementarity between the binding approach and the functional approach, and I think ideally that researchers should be doing both of those techniques, both to get biophysical constants for by binding assays, and also to get an assessment of how a drug works in a channel in the cell as well.
Okay, well, excellent. Well, thank you very much to all of our three speakers today, Dr. Luke Armstrong, Dr. Jake Isaacs, and Dr. Jeff Reagan. We appreciate all your information and your ability to help us out by answering a number of questions. And I'll just remind everyone again, if you could fill out the survey, it's in the chat box, that would be great. And we look forward to hearing back from our panelists on future events. Thanks, everyone.